The opinions expressed by private persons during the public proceedings of the Federal Interagency Committee on Indoor Air Quality are solely those of the speakers. The United States government and EPA do not endorse commercial products, services, or enterprises. Any mention in the CIAQ proceedings, meeting minutes, or presentations of a particular entity, product, or service is for informational purposes only. Such mention neither implies nor constitutes any endorsement or recommendation by EPA or the CIAQ member departments and agencies. Thank you.
Okay, we are very excited about our topics today. The first topic that we will be discussing is the management of Legionella in water systems. And this is being given by Dr. Laura Ehlers, who is the Senior Staff Officer at Water Science and Technology Board at the National Academy of Sciences. She received her BS from California Institute of Technology, majoring in biology and engineering and applied science. She's earned both an MSc and a Ph.D. in environmental engineering at Johns Hopkins and has been at NAS since 1997, where she served as the study director for more than 25 committees. Dr. Ehlers.
Thank you, Lorene, thank you for inviting me to talk about this recently completed report of the National Academies called Management of Legionella in Water Systems. I was the study director for the report. I'll give everybody a little bit of background, if we can, on legionellosis to bring everybody up to speed. So legionellosis is a medical condition caused by the bacterium Legionella, and about 80 to 90% of cases that are reported to the CDC have been specifically linked to Legionella pneumophila. There are two types of legionellosis. The first and more severe type is Legionnaire's disease. It causes pneumonia, and about 3 to 33% of the cases lead to death. What we know about Legionnaire's disease from the reported cases to the CDC is that about 96% of the cases are sporadic and that they're not associated with an outbreak. And about 70% of these cases are community acquired. In other words, they're not acquired from a hospital setting or from travel. The second type of legionellosis is a less acute, milder form of an upper respiratory tract infection called Pontiac fever, and it tends to resolve spontaneously and very frequently goes undiagnosed. So what are the risk factors for developing legionellosis? Age is a major one. Older people over 60 tend to be more susceptible. Males have a higher risk than females. Smoking or other chronic lung conditions can make you more susceptible. And finally, if you're immunosuppressed for any reason, you're much more likely to be at risk for legionellosis. So these two, the table and the graph, give us some idea of the extent of the problem. Right now, the CDC estimates that waterborne diseases cost about $1 billion a year, and almost half of that goes toward Legionnaire's disease, as you see in the table to the left. Not what you might more commonly think of as waterborne infections like cryptosporidiosis or giardiasis. The graph on the right shows us how things have changed over time in terms of the source of drinking water-associated outbreaks. Legionnaire's is now well over half of all outbreaks, and it's become more prevalent over time. It didn't even used to be, obviously, on anybody's radar screen. So this is a disease that's becoming more frequently discussed, heard about. I'm sure everybody has a Legionnaire's disease outbreak that they're familiar with. There was one recently in Atlanta, one in North Carolina. And so it's becoming a much more urgent health issue than it has been in the past. I think the reason that this topic is of interest to this group of stakeholders is because of how Legionella is transmitted. We inhale the bacteria when it exists in contaminated aerosols. And there are lots of common sources of Legionella bacteria in the built environment, it turns out. So showers and faucets that are connected to building plumbing systems can be areas where you find Legionella. They grow in cooling towers, which I don't have to explain to this group of people, which is fantastic. Many audiences don't really know what cooling towers are. Hot tubs and decorative fountains have also been found to be sources of Legionella bacteria. And so there are really two factors that determine whether something is a source. It's whether the bacteria can grow in that environment, and then whether that environment produces aerosols that can be inhaled and taken into the lungs. By the way, I'll just mention that this differentiates Legionnaire's disease from just about every other waterborne disease, most of which are of enteric origin and are ingested, not inhaled. This list of outbreaks is here just to kind of help one appreciate that outbreaks occur all over the world. They can often have hundreds of people who get infected, an average mortality rate of about 10%, and the sources can be highly variable. There are examples up here of whirlpool spas that led to an outbreak, several cooling tower examples, wastewater aeration ponds even, the Flint, Michigan outbreak, which was because of water supply changes, the Quincy, Illinois State Veterans Home situation, which was because of contaminated premise plumbing. So there are many different sources of Legionnaire's disease, and it has a significant amount of fatality associated with it. So where are we about 40 years now since we discovered Legionella pneumophila? We've learned a whole lot about the bacteria's ecology and biology, but we're not really all that good yet at preventing Legionnaire's disease. And we have also developed many different methods for monitoring both the disease incidence and the presence of the bacteria in water samples, but treating those water systems to reduce colonization by the bacteria is not particularly well understood. So kind of given that backdrop, in late 2017, the National Academies convened an expert committee to review the state of science with respect to Legionella contamination and write a report. And on the next slide, you'll see the bullets that kind of constitute what we call the statement of task. These are the questions that the committee was asked to address during its tenure. We were asked to talk about the ecology of water supplies as it relates to Legionella, talk about how can diagnosis of Legionnaire's disease be improved, what are the primary sources of human exposure to Legionella and why do they grow in water systems. A large portion of what we did was devoted to this quantification bullet in the middle. What do we know about concentrations of Legionella in water systems and whether there's like a minimum concentration above which you're going to see disease? And what do we know about the prevalence of Legionnaire's disease and how have these things changed over the last 20 years? The fourth bullet is about prevention and control strategies for preventing Legionella from amplifying in a water system. And then the final bullet was really an attempt to say, what are the guidelines, the regulations, the codes that might be protecting the public from Legionnaire's disease, and how could these be improved upon to better protect the public? Next slide, you'll see a picture of the committee that was constituted to do the study. For those of you unfamiliar with the way the National Academies works, all of these people were volunteers, and over a 2-year period, they met 6 times and wrote this report. The expertise on this committee was very broad enough to consider all of the 5 bullets in the statement of task that you just saw. Joan Rose was the chair of the committee. She's from Michigan State University and a member of the National Academy of Engineering. There were medical doctors on the committee, engineers, microbial ecologists, people who operate buildings. It was a really fantastic group of people to work with. Unfortunately, because of the way the Academies works, there were several experts who we would have liked to engage, but we couldn't because they were involved in litigation. You probably are aware that Legionnaire's disease is a very litigious topic with lawsuits in the millions of dollars for individual cases. And if people are involved in litigation, they couldn't serve on our committees. So we had to make use of their expertise in other ways by asking them to give presentations and things like that. So I'm not going to spend too much time talking about the introductory chapter. I've already kind of touched on many of the things in it. I just want you to focus on the slide, the picture, which shows that Legionella, it turns out they exist naturally in lots of aquatic environments and in soil and have never been a problem until they got into the built environment. And that happened because, we extract water for drinking water purposes, so you can find Legionella in treatment plants. Then the water enters buildings, if you follow the black arrows, and we find growth niches for Legionella there in hot water heaters, in cooling towers, in humidifiers, fountains, hot tubs, even in residences, something as simple as a shower head or a faucet. And we also find Legionella in wastewater treatment plants. So it turns out that the built environment is very ecologically favorable to Legionella because of the temperature and the lack of flow. So this is a problem that we've created for ourselves, and we have to figure out how to deal with it. So I'm going to talk briefly about Chapter 2. It's a real science-heavy chapter. It tries to help us better understand the biology and ecology of the organism and what we don't know. What we do know, though, is that there are 61 species of Legionella, and half of them are pathogenic. I've listed a couple of them at the bottom. The one we always think about is Legionella pneumophila, because it turns out that the way we diagnose Legionnaire's disease is using a test that only picks up Legionella pneumophila. But there are 30 pathogenic species, and it depends on where you are in the world. If you're in Australia, you're much more likely to get sick from Legionella longbeachii than you are from Legionella pneumophila. And I want to just touch on the complexity of the ecology so that you realize why this is such a tricky issue. So it's not just bacteria that are in the water, that then the water gets aerosolized and you breathe it in. Legionella live inside amoeba. They are parasites of amoeba. So in that lower right-hand corner, you see a gray blob. That's an amoeba cell. And inside there are different forms of Legionnaire bacteria. Some of them are replicated. Some of them are what we call the transmissive form. They have a little flagellum. Some of them are what we call the mature infectious form. And Legionella bacteria can also form what we call a viable but non-culturable form, which is a kind of a cyst-like form of the bacteria that actually you can't grow it, but it can retain its infectivity. We're not really sure if Legionella form these VBNC-like cells, as do Vibrio and some other bacteria, but it remains to be an open research question. So it's complicated because, first of all, they live inside amoeba. And then if you look at the pipe on the top, it's even more complicated because the amoeba grow by eating biofilm bacteria. Biofilms are these kind of slimy surfaces that coat the insides of pipes, and any wet surface for that matter. And so what you see there in the schematic are that the amoeba are inside the biofilm eating bacteria. They can detach from the biofilm. They can form extracellular vesicles. And the bacterium can exist in all of these various forms of the amoeba. So the idea is just to kind of present that there's a very complex situation, and controlling Legionella might actually be more about controlling the amoeba and the biofilms and all these other things going on in the environment, in addition to controlling the bacteria themselves. I'm going to just jump to some conclusions from this chapter. Obviously, I've just mentioned that the distinct Legionella cell types are very different. They might have different infectivity, different viability, different resistance to treatment like disinfection, and these things need to be taken into account. We don't know yet if Legionella enters into this viable but non-culturable state, and that remains to be an open research question. All the studies we've done so far have been really on Legionella pneumophila, and we have 30 other pathogenic species we need to know more about. The fourth bullet gets to the idea of better understanding the protozoan host of the Legionella and whether we could control Legionella infections by controlling the protozoa. And then finally, we don't really know how these bacteria cause Coniac fever. We don't know whether aspiration is a major component to disease. That's where you're inhaling food that you've ingested. We also don't know anything about how Legionella survive in aerosols. So these are some of the open science questions that the committee felt was important to highlight for future research. I'm going to jump to chapter 3, which is a data-heavy chapter where we were looking at two types of data. We were looking at data on incidence of Legionnaire's disease and also data on Legionella concentrations in water systems. So these two big types of data sets, we did some analysis of both of them to try to come up with some idea of how big of a problem do we have here. So let's go to the next slide. What you'll see, I'm not going to talk too much about methods in this particular presentation. I'll just mention that the methods for measuring Legionella are rapidly evolving. And there's a lot in the report about methods and the fact that it's very hard to compare method to method. Some are molecular methods, some are culture methods, some are urine antigen tests. But there's a lot of work to be done to be able to compare across methods. So the graph on the right is pretty alarming. It suggests that the reported rate to the CDC has increased 5 times in the last 17 years. And it's not much better in the European Union. That's the red line. They've also seen an increase in Legionnaire's disease over this time period. But the committee and most professionals know that actually the reported rates are an underestimate. So they tried to conservatively estimate a true incidence. So on the left, what you see is that the committee estimates that there are actually between 52,000 and 70,000 cases of Legionnaire's disease in the United States every year, which is 10 times higher than the reported rate that the CDC publishes. And this estimate, which the committee believes to be conservative, is actually supported by a lot of other independent studies. So I think, people might say, is that a large number, a small number? 52,000 to 70,000 new cases a year of a disease with a 10% fatality rate is significant. So the other large data set that the committee evaluated were the environmental monitoring data from around the world, people measuring Legionella concentrations in water systems. And they compiled, it's very hard to compile all these data. Most of it can't be compared to one another. People didn't use the same methods. They had the samples were taken differently, but we were able to compile a lot of big data sets. 8 are shown on this graph. And the green data sets were data that were measured during routine sampling, whereas the red and the yellow data sets were collected during outbreaks. And what the committee felt when they looked at these data is that there seems to be a break around 50,000 colony-forming units per liter. Or below this number, that you might detect that during routine monitoring, but above that number, you're in an outbreak situation. So they decided that this number, 50,000 CFU per liter, should be considered an action level or a concentration high enough to warrant serious concern and trigger remediation. Recognizing that, if you had a group of at-risk individuals like immunocompromised people in a hospital, then you might want to have a lower action level. 50,000 CFU per liter is actually, it's a moderate to high number. It's an alarm bell. If you measure that in a building, you should be extremely concerned that there is going to be an imminent outbreak. That's what that number means. This is the first time, to the committee's knowledge, that anybody has ever compiled environmental monitoring data on Legionella and attempted to see if there was some kind of threshold, a numeric threshold. And I'll come back to how we could use that numeric threshold later in my talk. The last slide in this chapter has to do with quantitative microbial risk assessment. So 50,000 is a high number, and you would be very concerned if you measured that. But what about if you just want to know, is my hospital contaminated with Legionella at some level that presents a risk? So quantitative microbial risk assessment can be used to help better understand the risk of, it depends on what your acceptable risk is, 10 to the minus fourth or 10 to the minus sixth. These are common values of acceptable risk for various situations. And I've presented 2 here. But QMRA is ready to say we have enough information on the processes for us to be able to associate these low concentrations with a low acceptable risk level for water coming from a faucet, a toilet, or a shower. Right now, QMRA really can't be done for cooling towers because the mechanisms by which an organism would leave a cooling tower and get into the lungs is just not well enough understood. But for these particular processes, we can say that these low levels are likely to be associated with low risks, and we can put numbers on both of them. So after dealing with all of this, I'm going to talk just about a few of these conclusions and recommendations. The ones that I've already talked about, I'll skip, but I will say that number 2 there, I talked in the beginning about how 96% of cases are sporadic, right? And I've shown you a lot of data, but much of it is data from outbreaks. The only way we're ever going to understand what causes sporadic Legionnaire's disease is going to be through a well-funded population-based study involving multiple jurisdictions. The committee also recommended that there be a regional center of excellence for Legionella to help strengthen local and state health departments when they do investigate cases of Legionnaire's disease. They don't usually have the time, the money, or the expertise to figure out where someone contracted Legionnaire's disease. Was it from their shower? Was it from a building they walked by and they inhaled aerosols from a cooling tower? So there's just a big vacuum of information that needs to be filled. I'm not going to talk about the other items. I'll go right on to Chapter 4, which is a chapter that we devoted to control. So if you have a building and you're trying to prevent it from getting colonized by Legionella, or you have Legionella there already, what do? There are some, very main control strategies that are shown in this a little bit complicated figure. They're in the bubbles. So the 4 main control strategies that we talked about in this report are the use of disinfectants, choosing plumbing materials, elevating the temperature, or hydraulic design and management. There were also 3 kind of less used strategies that we talk about, including nutrient limitation, control of aerosols, and distal design. And for a given building type, many of these control strategies could be used. You might choose 1 or 2. It really depends. Shown on the next slide is a table where all of the control strategies are listed on the left-hand column. And across the top, you see 9 different building types or engineered systems or devices. And what you'll see is that, if you're looking at, for example, cooling towers, there are some of the strategies that will work and others we're not sure about. If you see a check mark, it means that we have evidence that that control strategy works in that building type. Question marks, we think there's some potential for the control working, but there could be limitations. If you see nothing, it means we don't know. And if there's an X, those are situations where it suggests that the building type actually will prevent a strategy from working. And those are all under the green building column, and I'll talk to you about that when I get to the end of this chapter. And I'm not going to exhaustively go through why every control strategy works in every building type. I'll just go ahead and kind of skip to the punchline, which is that some of these control strategies are fundamental. The most important one, for which there is the greatest evidence, has to do with temperature. Hot water heater temperature must be above 60 degrees C, and the hot water temperature to the distal points of plumbing systems should exceed 55 degrees C. There is ample evidence in many large buildings that keeping the temperature of the water outside the preferred growth range of Legionella is a very effective strategy. And when I say outside the preferred growth range, they prefer 25 C to 43 degrees C. So you've got to keep the cold water below 25 and the hot water above 43. And the studies suggest that these temperatures of 60 degrees C and 55 degrees C will prevent Legionella from growing in your building. So that's a very important recommendation that will come up again later. With respect to disinfectants, the committee found that monochloramine is better than free chlorine at controlling Legionella risk, both in water distribution systems and in the plumbing of buildings. And the reasons for that are very interesting and have to do with the amoeba host, but we're not going to get into that. A related issue is that, you might all be thinking, the water comes into my building is from a public water supply, and they chlorinate their water supply, so I'm protected, right? That's actually not true. We don't know how well the disinfectant residual from a distribution system actually penetrates into most buildings. Especially large buildings, you can probably be pretty sure that there is no disinfectant residual. And this is an area of research that really desperately needs to have some attention paid to it. A fourth important point from this chapter is that there's a lot of information that we give that could be very relevant to individual homeowners, especially people who are immunosuppressed. We think that one of the sponsoring agencies needs to create some formal guidance to homeowners for how they can protect themselves from Legionella. A fifth important issue from this chapter is that low-flow fixtures should not be allowed in hospitals and long-term healthcare facilities. These fixtures are known to be very favorable for Legionella growth. They have low to no flow. Water stagnates. They have no disinfectant residual. Any kind of temperature control that you might have been trying to achieve will dissipate. And they simply cannot be allowed in situations where high-risk occupants, like immunosuppressed people, could be exposed. We also talk in this chapter about new designs that are needed for cooling towers and humidifiers where you have fewer options. And then finally, we spend a lot of time talking about why green buildings and water and energy conserving features have made things worse. These buildings were not designed with Legionella in mind, and many of their features actually create favorable growth initiatives for Legionella. So using energy conservation as an example, frequently you are told to reduce the temperature in your hot water heater to save energy. And what you're doing is you're bringing your hot water heater into the preferred growth range for Legionella. Water conservation is a similar problem. As we conserve water, we reduce the flow inside the building. The water residence time goes up. Whatever disinfectant residual you might have had will dissipate. Whatever temperature control you might have had will go away, and suddenly the building plumbing is much more ripe for Legionella growth. So these buildings need to be redesigned. They need to be retrofitted so that these problems can be eliminated. I'm going to move to the last chapter now, which is about policies and guidelines. So I mentioned a little bit already that if you have a water supply that's a public water supply coming into your building, you might think, I'm good. What the water utilities have to do to respond to the Safe Drinking Water Act is they have to provide you with a disinfectant residual up to the point where the main distribution system connects to a building, which is at the service line. And that is where their responsibility ends. What happens once the water is inside a building, inside the premise plumbing, nobody really knows, but there is no evidence that disinfectant residuals persist within these buildings, especially as buildings get larger and plumbing gets more complicated. There are lots of reasons for this. The pipes have a much higher surface to volume ratio and are more amenable to biofilm growth. The water age is probably a lot larger and much more highly variable. So contrary to what you might think, the upshot of all this is that the Safe Drinking Water Act does not protect people from Legionella. So what do we have in the United States right now? We have kind of three basic options. There are a small subset of buildings for which there are Legionella regulations, and these regulations either require a water management plan and or monitoring of the building water system for Legionella. So the first three bullets, you see the blue bullets, both things are required. So for healthcare facilities in New York State, cooling towers in New York City and New York State, and healthcare facilities within the Veterans Administration system, they have to have both water management plans and monitoring of their water systems for Legionella to see if those plans are working. The fourth bullet that's green, hospitals and healthcare facilities that receive Medicare and Medicaid funds are required by the CMS memo of 2017 to have water management plans. So that's one group of buildings that is protected in a sense, but it's, not a large number of buildings. The second category is if a building owner or manager decides to voluntarily create a water management plan for a building, and there are many guidance documents that we reviewed, ASHRAE 188, NSF International 444. There's a good one from the AIHA. So you can voluntarily have a water management plan, and some building owners and building types have been proactive in this way, particularly hospitals with immunocompromised populations and some hotels. Then there's a third category, which is everybody else in every other building, which really can only be protected from Legionella through the application of building and plumbing codes, which, as we discuss in the report, are totally inadequate for that purpose and not even written with Legionella in mind. So that's the kind of the state of things. If you go to the next slide, I give just a very brief overview of what is a water management plan. And what we're getting at here is that these are the plans that people establish. It's a document where you say, here's what my water system looks like. Here are where the potential hazards could exist, where Legionella could potentially grow. Here are the control measures I'm going to use to prevent that growth. Maybe it's elevated temperature, maybe it's biocides. I'm going to monitor some parameters. It may not be Legionella, but I'm going to do some monitoring to determine if control strategies are working. And then I have to confirm that everything is being implemented properly and working as it's supposed to, and I'm going to document everything. That's a water management plan. And that right now is really only required of hospitals that get CMS funding and of hospitals that are in the VA system, and then everything in New York State. So one of the interesting things we tried to do was say, okay, is the United States unique? Is it, is everybody kind of not really doing much right now? Well, that's not the case. So you'll see in this table that we have evidence that in Western Europe, Canada, and Australia, there are actually a lot of regulations for Legionella. Some are provincial, some are national, and they cover all different types of buildings and devices. They sometimes specify different preferred treatments, whether it's temperature control or biocides. They all have monitoring thresholds, which means they all require that these buildings get monitored for Legionella, and they specify a value above which certain actions have to be taken. Things are pretty different in these other countries, and we actually had representatives from all of these countries come and talk to the committee about whether or not these regulations have had any effect, number one, on environmental concentrations of Legionella, and number two, on Legionnaire's disease rates. And while nobody was willing to say that the regulations have had any effect on Legionnaire's disease rates, because we know from that previous graph that rates in the EU are going up, three of these representatives, two in Canada and one in Germany, do have significant evidence that these regulations have led to lower environmental concentrations of Legionella, especially in cooling towers. So what can we do about this? What the chapter ends up saying is in the United States, we've got virtually nothing except for a small subset of buildings. How can we incrementally better protect the public? Number one, we would expand the CMS memo so that all those hospitals that we say have to have a water management plan, they also have to monitor for Legionella in their buildings to see if their plans are working. Number two is to register and monitor cooling towers, and I mean monitor for Legionella. This is low-hanging fruit. It's been working well in Quebec. It was done in Garland, Texas. It's happening in New York City. This would be something that would be relatively easy and have a large impact on our ability to figure out where Legionnaire's disease cases are coming from. Number 3 is a very difficult recommendation, but one that we thought was really important to make. We think all public buildings, hotels, businesses, schools, apartments, everybody should have a water management plan. You should be able to walk into a hotel and ask to see a copy of their water management plan. We didn't recommend that all public buildings monitor for Legionella. We thought that would be too difficult, but we do think they should have a water management plan because at the very least, it makes the building owners and operators aware that their building could potentially harbor these bacteria. Number 4, we were so concerned about the efficacy of the temperature control that we felt like it needs somehow to be codified. So we think that a temperature of 60 degrees C at hot water heaters and 55 degrees C to the distal points of plumbing systems needs to be codified either in building or plumbing codes or added, for example, to the CMS memo or put into the VA directive or something. And 55 degrees C does not mean that that's what would necessarily come out of the tap. You can always add a thermal mixing valve to lower the water temperature and prevent scalding. But you need to have 55 degrees C coming to the distal point. That's very important. And the final thing we recommend is that there be a minimum disinfectant residual throughout public water supply systems. Right now, EPA allows a few samples to be 0, and we felt like, there really should be a minimum residual, measurable residual throughout all systems. We don't prescribe what that number is, but we have some suggestions. And we also thought that utilities should maybe start monitoring for Legionella to see if it's there. These 5 recommendations, by the way, they're any one of them, making progress on any one of them would lead to lowering public health risk. And we realized that some of them are really difficult to implement, that they would be costly to implement, that there would be timing issues. We try to talk about the training and education needed to carry out these recommendations. A couple of our recommendations ask for new monitoring of Legionella, which has really scared a lot of people. People don't want to monitor for Legionella. They don't want to know if there's a problem there. What do they do if they detect it, So there needs to be guidance on how would you be interpreting monitoring results. And this is where you could go back to the 50,000 CFU per liter threshold or one of the lower numbers that comes out of the quantitative microbial risk assessment to help interpret the monitoring data. Finally, I just want to suggest that as the right-hand side of the slide suggests, there are all these stakeholders, and it's really true. Legionella doesn't fall squarely on the shoulders of any one federal agency or entity. All of these groups of people play a role, from clinicians who diagnose it, to people who operate buildings, to the federal agencies that have some tiny piece of the pie, whether it's the Department of Veterans Affairs or EPA. And it's going to take a group effort. So what we're doing is trying to educate as many different groups of stakeholders as possible in the hopes that you will look at the report and say, that's something that I could act on and try to move some of these recommendations forward. Go to the last slide and you'll see the URL for our report. It's currently in pre-pub version on our website. I welcome you all to download it, take a look at it. There are some derivative products that are shorter and easier to digest than the full report, which is a couple hundred pages long. There will be a final color version available at the beginning of 2020. And I thank you, everyone, for your attention, and I'm happy to take any questions.
So our first question, for the wastewater data in your slides, what approximate distance from the pond was the concentration reported from?
Oh gosh, I don't know, but I'd be happy to go back and peruse the papers. The wastewater data are very interesting and kind of isolated right now to Northern Europe, but they have found that it's being aerosolized above aeration ponds and that people downwind of those ponds have gotten sick. But I'd be happy if you tell me your contact information. I'd be happy to make sure you get the appropriate citation.
I will pass that on after the meeting. For our next question, would a recirculating domestic hot water system reduce Legionella bacterial growth potential?
What's the growth potential in a recirculating hot water system? It really depends on what temperature you're maintaining the hot water system at. The committee would suggest that you need to have the hot water heater at least 60 degrees C, and you need to have at least 55 degrees C in all of the recirculation loops and out to the distal points. And if those criteria were met, then you would have very low, if any, Legionella growth potential. The problem is that, many plumbing codes and building contractors, they would never set a hot water heater at 60 degrees C, which is the same as 140 Fahrenheit. That's mainly for scald concerns. So the committee has a table in the report that tries to demonstrate how one could balance scald risk along with Legionella growth potential risk. And they felt like these two temperatures of 60 and 55 can be balanced. You can balance those two risks at those two temperatures. That is not to say that if you're in a hospital setting where you have a vulnerable population, as you may at a VA hospital, that you wouldn't want to use thermal mixing valves to lower the temperature so that you don't have 55 degrees C at the tap. That's those are considerations that have to be made on a case-by-case basis, for sure. But if you can achieve 55 degrees C to the distal point, you will significantly eliminate any Legionella risk.
Thank you. For our next question, this person asks, thank you, Dr. Ehlers. You stated that the SDWA does not protect against Legionella. Is there anything in the works legislatively to set protections?
No, there isn't. There are parts of the Safe Drinking Water Act that mention Legionella and that assume that there will be protection afforded by the disinfectant residual. And I think that, compliance with the Safe Drinking Water Act requires that utilities provide that residual up to the service line. The problem is that the Legionella are growing in premise plumbing of buildings, which is beyond the responsibility of the utility and, frankly, beyond the reach of the Safe Drinking Water Act. So there is nothing that we know of that is in the works. There were some smaller side issues that affect Legionella and affect whether or not a building water system would have to be categorized as a drinking water provider in order to put secondary treatment within that building. But I think those are kind of peripheral issues, and while they may solve some of the problems, it's not like they're any kind of legislative fix to the issue of Legionella contamination of building water systems. Unfortunately, Legionella, because it's, in these built environments that are outside the scope of EPA's regulations, they're outside a lot of different groups, kind of comfort areas. It's hard to know who should be responsible. The VA is a unique situation because they own and operate all of these buildings, and so they have been able to create protections through these directives where they require water management plans. But there is really no other kind of federal agency level protection that would be obvious for us to recommend, which is why those 5 recommendations at the end are kind of chipping away at the problem in various different ways. But none of them, you see that there were no suggestions that laws be rewritten or amended or anything like that.
Thank you. For our next question, this person asks, how long has it been since New York began requiring monitoring of Legionella?
Not long, probably just 2 or 3 years. And we spoke with the groups in New York that are collecting those data. And, it's a struggle because all of a sudden there are huge amounts of data from healthcare facilities coming into a state agency that probably doesn't really have the capacity yet to know what to do with those data and how to interpret the data. So it's a little bit of an experiment. I think that, probably at this early stage, the more important part of that regulation is that all of the healthcare facilities and hospitals have a water management plan, and that there's somebody in the hospital who is thinking about Legionella, thinking about how to protect patients from Legionella contamination. How useful the monitoring data will be at these early stages is not yet clear. But just in general, I can tell you that most buildings, we have no idea if there are Legionella in them. So just being able to have data and say, yes, it's here, or no, it's not, is extremely powerful. And we're just at the early stages of trying to figure out how to manipulate and analyze these data to come up with meaningful things to say about the extent of contamination.
Thank you. For our next question, for homeowners, would soaking/cleaning a shower head with a Clorox mix help?
So I'm going to balk just a little bit and suggest that for a homeowner, you should get the report. You should read Chapter 4, and there's a section there on residences that will provide helpful information. There are some basic principles that I think people can try to implement at home. The setting of the hot water heaters to a higher temperature is a major one, but that's something that you really have to be very careful with because you have to balance the higher temperature against the potential for scald risk. You know, who's in your home, who might be exposed to scalding risk. So that is an individual choice. There are flushing issues that I think could be very useful for homeowners, such as flushing their plumbing when they come home from a vacation. Most people don't even think about the fact that when they're gone for a month on a vacation, their water has been sitting stagnant in their pipes while they're gone. If there was a disinfectant residual, which we don't know if there was, it's surely gone by now. And if there was any kind of temperature control afforded just downstream of the hot water heater, that's also probably gone. So flushing is kind of a very practical, easy to do thing that I think homeowners could exercise. Exactly what the frequency of flushing is, that's why we need some formal guidance from the CDC or from EPA to help homeowners through these issues. That section of the report also talks about the use of distal devices such as point-of-use filters, which mainly are for faucets but are now being designed for shower heads. These devices can protect people and would be useful in homes of immunocompromised people, but they can also be sites where you could get regrowth of Legionella. So there are tricky issues on both sides. Your specific question about cleaning shower heads with Clorox bleach. We recommend cleaning devices, particularly humidifiers in homes. For sure, I don't think you can go wrong with cleaning a shower head. It's certainly not going to make the problem worse, and it might make the problem better. But if you have Legionella contamination in your hot water heater, you're just going to recolonize the shower head. If your hot water heater is sitting there in a happy growth range for Legionella, it won't end your problem. So I encourage people to look at Chapter 4 and to try to pick out the pieces that you think would be useful in your home. There are nuggets there, but the committee did not feel like it could make formal recommendations to homeowners, which is why they've asked one of the federal agencies to do so.
Thank you. For our next question, this person asks, are there any resources unique to California?
Gosh, I don't think so. Legionnaire's disease is everywhere. It's particularly acute in the Northeast, but it's going to become more of a problem in the Southeast and in warmer places where cold water is never very cold. And one of the things that the report doesn't talk about is how to keep the cold water cold. You have many, many fewer options. And in places like the Deep South, where the cold water system is above 22 degrees Celsius, there could be an increase in Legionnaire's disease cases as a result. Some of that is being counteracted by moving toward monochloramine as a disinfectant in those public water supplies, and we know that monochloramine is more effective against Legionella than chlorine. But other than those regional differences I've discussed, we didn't talk specifically about anything unique to California or the West Coast.
Thank you. Lorene, over to you for in-room questions.
Does anyone in the room would like to ask a question? Lisa?
Yes, thank you for your very informative presentation. I had a question about hard water versus soft water utility areas. Do you see a difference in Legionella and those kinds of systems?
It's not in the report, and I don't recall any committee member discussing differences between hard and soft water, so I'm afraid I can't say. I think if there were differences, that the committee would have discussed them. So I would hazard a guess to say there are none, but it's not discussed in the report.
Okay. Do we have any last question on the chat room?
Yes. This person asks, is there guidance for a water management plan?
Very much so. You can find excellent guidance from a number of different sources, including ASHRAE 188. That's the Association of, American Society of Heating and Air Conditioning. I'm going to get it wrong. ASHRAE, if you look up ASHRAE 188, if you Google it, you'll find their most recent guidance, which I know is being updated. The Association of Industrial Hygienists has a good guidance document from 2016. The Veterans Administration Directive is not a particularly user-friendly document, but it's a very comprehensive document about how to create a water management plan. I would start with ASHRAE 188. It's a short document, and it has kind of some appendices that can help you if you have a specific building type, like a cooling tower or a healthcare facility or something like that. You can learn a lot from, it's only about 25 pages long, so that's where I'd start, is ASHRAE 188.
Okay. Well, thank you.